Welcome to Biotanemy.
Mitosis. Mitosis is a type of nuclear division in which a parent cell divides to produce two genetically identical daughter cells, each having the same number of chromosomes as the parent cell. It ensures growth, repair, and asexual reproduction in multicellular organisms, and occurs in somatic cells. Different phases of mitosis are: 1. Prophase. In prophase, the chromatin condenses into visible chromosomes. Each chromosome has two sister chromatids joined at the centromere. As we discussed in the earlier video about the cell cycle, the cell enters M phase by the activation of the Cyclin B, CDK1 complex becomes fully activated. Condensin complexes 1 and 2 are phosphorylated by Cyclin B, CDK1, enabling them to bind chromatin. Topoisomerase 2 alpha resolves DNA supercoiling and tangles between sister chromatids, leading the chromatin to condenses into distinct chromosomes, each with two sister chromatids joined at the centromere. Centrosomes duplicate in S/G2, but mature in prophase. Aurora kinase A and polo-like kinase 1 phosphorylate centrosomal proteins, leading to nucleation of microtubules. Gametubulin and associated proteins at centrosomes initiate spindle microtubule growth. Spindle fibers begin forming between centrosomes, moving to opposite poles, setting the stage for nuclear envelope breakdown, which is completed in Prometaphase. Cyclin B, CDK1 phosphorylates nuclear lamins A, B, C, leading to disassembly of the nuclear lamina. Nuclear pores disassemble, making the chromatin accessible to spindle fibers. Other regulatory events that occur during this phase are: histone H1 kinase activity via CDK1 modifies histones, aiding condensation. Aurora kinase B, a part of the chromosomal passenger complex, begins to localize to centromeres, preparing for kinetochore microtubule attachments in Prometaphase. The mitotic spindle begins to form from centrosomes. The nuclear envelope starts breaking down.
2. Prometaphase. The nuclear envelope completely disappears. Spindle fibers attached to kinetochores and chromosomes begin moving towards the equator. Prometaphase begins immediately after prophase, once the Cyclin B/CDK1 complex activity rises sufficiently high. This kinase phosphorylates nuclear lamins, A, B, C, and nucleoperins like NUP98. This results in the release of the chromosome into the cytoplasm by nuclear envelope breakdown. The spindle microtubules now have free access to chromosomes. Kinetochores, the protein complexes assembled on centromeres, serve as docking sites. Microtubules extend from spindle poles, randomly searching for kinetochores, and they capture chromosomes once contact is made. Initial attachments are often faulty. The enzyme Aurora B kinase, which is a part of the chromosomal passenger complex, senses tension. If wrong attachments form, Aurora B destabilizes them, allowing reattachment. Once microtubules attach, chromosomes don't stay still. They oscillate back and forth. This movement continues until all chromosomes align at the metaphase plate. Balance is achieved between the pulling forces of opposite spindle poles. By the end of Prometaphase, kinetochores of each sister chromatid are attached to microtubules from opposite poles. 3. Metaphase. In this phase, the chromosomes align at the metaphase plate. Spindle fibers from opposite poles attach to the kinetochores of each sister chromatid. As chromosomes are actively moved back and forth by a tug-of-war between kinesin, 7 or CNP, and dynein. These forces balance out, causing chromosomes to line up at the cell equator or metaphase plate. The SAC checkpoint will be active at this point to ensure proper attachment and tension of the kinetochore. The details of the SAC checkpoint are explained in the previous video. Please watch using the link given in the icon and description box. The kinetochore microtubule attachments are stabilized by enzymes like 1. Kinesin 13 or MCAK, which depolymerize the incorrect microtubule attachments. 2. Aurora B kinase continues to correct the improper attachments until full biorientation is achieved. 3. PP1/PP2A phosphatases balance the Aurora B activity once correct tension is achieved. Once all kinetochores are attached and under tension, sacs dissociate. CDC20 is freed and activates APC/C. APC/C will then target securin for degradation in the next phase, allowing separase to cleave cohesin. 4. Anaphase. In anaphase, sister chromatids separate at the centromere. They are pulled apart to opposite poles by spindle fibers. Each chromatid is now an individual chromosome. Before anaphase starts, the spindle assembly checkpoint ensures all kinetochores are properly attached to spindle microtubules. Once satisfied, the mitotic checkpoint complex, which consists of MAD2, BUB-R1, and BUB-3 proteins, is disassembled. This allows anaphase promoting complex or cyclosome, activated by CDC20, to function as a ubiquitin ligase, and helps degrade securin. Securin normally inhibits the protease named separase. Once securin is degraded, separase becomes active. Separase cleaves cohesin, mainly the SCC1/RAD21 subunit, which was holding sister chromatids together. This cleavage triggers the physical separation of sister chromatids. Once sister chromatids are free, kinetochore microtubules shorten. Depolymerization occurs at kinetochores and spindle poles. Chromatids are pulled towards opposite poles. While anaphase A pulls chromatids apart, anaphase B elongates the spindle by sliding the interpolar microtubules apart. Kinesin 5, also known as EG5, pushes overlapping microtubules apart. Dynein anchored at the cortex pulls centrosomes toward opposite cell sides. Degradation of Cyclin B by APC/C reduces CDK1 activity, which is essential for progressing into telophase and cytokinesis. Low CDK1 activity allows nuclear envelope reassembly and chromosome decondensation later. 5. Telophase. At this phase, chromosomes reach opposite poles. The nuclear envelope reforms around each set. Chromosomes decondense into chromatin, and spindle fibers disassemble. By the end of anaphase B, sister chromatids are fully segregated to opposite poles of the cell. Motor proteins have completed their work, and dynein pulls chromosomes toward the spindle poles along microtubules. Kinesin 13 will help to depolymerize microtubules for final movement. The mitotic spindle microtubules are no longer needed, so they depolymerize. The key regulators in this phase are catenin and spastin, which help to sever and depolymerize microtubules. Cyclin B is degraded and decreases CDK1 activity, which is required to maintain spindle stability. The reduction of CDK1 activity triggers spindle breakdown. Nuclear envelopes are reformed now, and chromosomes get enclosed in two separate nuclei. Here are the events like fusing of nuclear envelope vesicles from the ER around chromosomes, reassembly of nuclear pore complexes, and the lumen proteins repolymerize to form the nuclear lamina. Chromosomes relax back into diffuse chromatin, preparing for transcription in daughter cells. The histones are dephosphorylated, removing mitotic marks. Chromatin remodelers reorganize DNA. By telophase, all SAC signals are gone because all chromosomes are properly segregated. The decrease in CDK1 activity and reactivation of phosphatases drives progression towards cytokinesis. 6. Cytokinesis. Division of the cytoplasm occurs during cytokinesis. In animal cells, cytokinesis occurs through cleavage furrow formation, beginning during late anaphase and telophase. Actin filaments and myosin II assemble just beneath the plasma membrane at the equator, forming the contractile ring. The ring contracts inward, creating the cleavage furrow. Myosin II motors slide actin filaments past one another, like a tiny muscle. The plasma membrane is pulled inward. The furrow deepens until the cytoplasm is pinched into two daughter cells. At the mid-body, membrane scission occurs. The plant cells, on the other hand, can't pinch inwards since the cell wall is rigid, so they form a cell plate. After anaphase, microtubules and actin filaments form a scaffold called phragmoplast at the equator. Golgi-derived vesicles carrying cell wall precursors like pectins, hemicellulose, and collose are transported along phragmoplast microtubules to the equator. Vesicles fuse via snare proteins to form the cell plate. The cell plate expands outward until it fuses with the parental plasma membrane, which develops into a new cell wall separating the daughter cells. That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.